Hi, my name is Vaughn Cooper. I'm a professor at the University of Pittsburgh and the Department of Microbiology and Molecular Genetics in the School of Medicine. I'm an evolutionary biologist and a microbiologist and a co-founder of our Center for Evolutionary Biology and Medicine. Thanks very much for the invitation to talk about viral evolution with you. So the goal of the talk is to ask, what has evolutionary biology taught us about SARS-CoV-2? And what, if anything, has SARS-CoV-2 taught us about evolutionary biology? Which leads to several questions, beginning with first, where did this virus come from? So what let the proverbial cat out of the bag and cause this pandemic? So to help explain, let's first consider this family tree of cats. This figure indicates a series of nested groupings among cat species. And at the far left, hopefully you can see that the lineage that gave rise to these big cats diverged from the domestic house cat lineage about 12 million years ago. Over on the right, notice that the snow leopard and the tiger are actually sister species that diverged from one another about 3.4 million years ago, meaning that snow leopards are more closely related to tigers than they are to leopards, which is pretty cool. In any case, we can use the same rationale to infer genetic relationships between different types of viruses isolated from different host animals. So if we now focus on the viruses that are related to SARS-CoV-2, we can build a family tree of coronaviruses. And so now what I've done is I've highlighted all the coronaviruses that are known to cause human disease. Now at the bottom, please see several coronaviruses, HKU1, OC43, and so on, that cause a common cold. And their nearest neighbors are viruses found in rodents, mice, and rats. But above, there are all the other coronaviruses that have caused severe epidemics in the last 20 years, MERS, SARS-CoV-1, and now SARS-CoV-2. All of these are closely related to viruses isolated bats. And you may notice this other creature, the masked palm civet, from which might have served as the intermediate species that gave rise to the original SARS pandemic. However, this civet was almost certainly infected by a bat recently beforehand, and more recent analyses have expanded the number of close relatives of SARS-CoV-2 to isolates from bats in China and Cambodia. So all the severe coronaviruses trace their very recent ancestry to bats, but it's possible that they passed through another animal immediately prior to infecting humans. But some of you might still be saying, okay, Vaughn, okay, it's related to bats, but where did this virus really come from? Well, fortunately, last month, a group of top evolutionary biologists, virologists, and epidemiologists published a forensic study of the epidemiological sources of the origin of the COVID pandemic. And they discovered numerous samples of SARS-CoV-2 clustered in just a few stalls in the Huanan seafood and wildlife market. And these cages has raccoon dogs and other various birds that are very plausible intermediate hosts of this virus. Some of these have even more recently to house different coronaviruses related to SARS-CoV-2. So with this evidence, it's extremely likely that one or more of these animals, perhaps in these cages, became infected with SARS-CoV-2 by a bat, was trapped, brought to the market, and then that served as the source of the infections of humans. These initial infections differed by one or two mutations, which went on to define sort of lineage A and B, and each of those went on to spread within the broader Wuhan community and then on globally. So an evolutionary biologist would then ask, so then why are bats the reservoir for human coronaviruses? And one explanation is that 20% of a mammalian species are bats. So it makes them a pretty sizable and mobile reservoir of viruses that could infect other mammals. The second is that bats have evolved a broad range of metabolic capacities that enable their success as fast-flying predators and their long lives. Bats can live several decades, so they have exceptional defenses against byproducts of their high metabolism. And the third is that their antiviral responses are actually dampened, ironically, through a pathway known as STING, so that their immune responses to viral infections are measured and not as damaging to bats. So they can tolerate and manage viral infections better than we can. Okay, so we know this novel coronavirus came from bats, like the other two coronavirus outbreaks. We know that there are many potentially dangerous bat coronaviruses from sampling bats in the wild. Our research over the last 2 years has shown that these viruses have even more capacity to recombine with one another to produce new lineages and to evolve greater pathogenesis. That's all clearer. How is it evolving in humans now? So the evolutionary changes with the novel coronavirus have been captured and analyzed beautifully by the group who built and run nextstrain.org, which provides near real-time visualizations of genetic differences evolving within epidemics like influenza, Ebola, and for the last 2 years, COVID-19. On the x-axis of this graph is time, and over the y-axis is the number of mutations. And so what this graph is showing over the timetable, over the roughly 30,000 nucleotides in the SARS-CoV-2 genome, and of about from about 3,200 genomes that represent the more than nearly almost 10 million virus genomes that are now available on global databases, we can basically retrace the evolutionary history of this epidemic. The average distance of a number of mutations from any virus sampled today compared to the original one is about 65. That original genotype from Wuhan is essentially extinct. It's been replaced by all of the new lineages and is only found in lab stocks for people studying the virus. Despite all of this genetic variation, we still consider this just one strain. We can look at these differences that are coded now by the different major named variants, these orange and red lineages of the Omicron variants, differently. And so here's now a variant-focused view of the same data showing sort of discontinuous changes where the big named variants that have captured our attention and have caused surges in the epidemic seem to emerge rather discontinuously. So you have a sort of sudden number of mutations producing the alpha lineage, a number of mutations giving rise to the delta lineage, and then a huge number of mutations tracing all the way back to fairly early on in the pandemic, back in mid-2020, from its most recent sampled ancestor, the Omicron lineage, which had a number of mutations. We can look at this, this also just by focusing on mutations in the all-critical spike protein that binds to our cells and allows the virus to infect. Mutations in this protein are important for escaping antibodies and for infecting different cell types in our bodies. Once again, you can see the evolution of spike mutations is discontinuous with the huge number of mutations found in Omicron. So why the changes in the variant lineages is not uniform is something that I'm going to return to in a few slides. So with all of this change, you might think, wow, the power of natural selection for this virus on this virus is quite high, but you may be surprised to learn that the power of selection on the virus remains relatively weak because of population bottlenecks. So each of us is actually infected by relatively few genetically different viruses. And although many mutations arise during each COVID-19 infection, the number of viruses that is transmitted to go on to form a new infection is effectively still quite small, on the range of only 1 to 10 viruses. So what I'm showing you is a data from this study by Lithgow et al., but also I've now analyzed some other data from Braun et al. From another major paper last year, showing a histogram of minority variant frequencies, meaning mutations that are at different frequencies, not at 100% within patients. And so the vast majority of new mutations that you detect within patients are rare. And then because they are rare, they fail to transmit. So, and just to summarize this point, the small bottleneck means that most mutations are born and die within a patient and fail to transmit. However, some of these mutations are lucky. And so when you have millions of infections every day, you have some number, thousands of lucky mutations that go on to form infection clusters. So this is a snapshot of the past wildfire season last fall out west. And you can see lots of different wildfires of varying size across the West. You can imagine that these are also like infection clusters, and then occasionally conditions are right so that some of them spread to form bigger and bigger clusters, forming uncontrolled wildfires. That selection is acting on more transmissible lineages that spread faster and faster and faster. And so I want to just enforce the idea that once lucky mutations that actually are fortunate enough to increase the fitness of the virus start to pick up a head of steam, and conditions in us, meaning incomplete vaccination, changing non-pharmaceutical interventions like masking and distancing and airflow happen, the lucky mutations can form these embers that are allowing them to spread, and they just start to rage out of control. And that's exactly what we've seen with Alpha and Delta, and now currently Omicron, BA1 and BA2 variants. So these uncontrolled epidemics are like evolutionary windstorms over smoldering embers of sort of hopeful mutations. So we now step back and now get back to the genome and look at, all these lineages that we're sampling by this massive effort in genome surveillance. We can ask what might be happening to allow the virus to evolve, to become better adapted. And one major signature of adaptation is convergent evolution, where the same mutations happen or the same traits evolve over and over and over again. So now I'm showing you different desert animals, and these animals show great examples of convergent evolution of a sandy colored pigment for camouflage, large ears to dissipate heat in the desert. These are classic convergent adaptations among desert animals. We can apply the same sort of logic by looking at the virus genomes, and you might be surprised to learn that evolution in the virus genome seems to be continuing to retrace many of the same steps. And so we published this very early on in the pandemic, and this has been borne out in a number of other studies, including an editorial that synthesized some research I wrote last year. And so I'd argue, even still, despite all of this variation, it's the similarities among these major variants that deserve a lot of our attention. And so in the middle, I hope you can see the many mutations in the spike protein captured by this wonderful resource called Covariants, produced by a collaborator of ours, Emma Hodcroft, that illustrate the many spike mutations in the Omicron variant that make it quite different from the previous variants. But we can also compare those two Omicron lineages, BA1 and B2, with Alpha, Beta, Gamma, Delta, and so on. And I hope you can see that they share these mutations in common. And experiments have gone on to show that these two mutations, actually a single deletion that takes away two amino acids, increases the rate of cleavage of the spike protein that's essential for it to actually activate and penetrate our host cells. They also share mutations in a particular site at 681, which is extremely important for expanding protease activity and higher virus loads. And in addition, they share a mutation at position 484 that is very powerful at reducing the binding of neutralizing antibodies. So natural selectin is acting on the same types of mutation arising over and over and over again in different lineages and causing them to rise in frequency as these embers become their own wildfires. You can visualize these same mutations on a 3D image of the 3D model of the spike protein, and they localize to particular regions of the spike protein that are functionally important. And so that really allows us to learn more about how the virus works and how it's adapting, how in turn we can design new therapeutics. So I previously said that I wanted to talk about the discontinuity in the rate of evolution. And one thing that it's become clear that we have to pay attention to is the role of these long-term chronic infections. So a major change with the spread of the pandemic is that more people who are immune compromised become infected. And it's long been known that immunodeficient individuals are susceptible to chronic infections of bacteria, viruses, and fungi, and so on. which can enable larger pathogen populations that persist over a longer period of times, which basically gives those pathogens more evolutionary leeway. And as Elaine Vitone, who wrote about this work that we've been a part of, put it, it's the virus is like throwing spaghetti at the wall, and every so often, although most of those mutations are useless, occasionally a good mutation happens and it sticks and it's able to transmit. So in a healthy individual, pathogen diversity is low and the number of spaghetti noodles that stick are very few. But in a immune suppressed person, the pathogen diversity is large and you may have many more mutations that ultimately stick. And so there have been many studies that have shown the power of the virus to an extent to which the virus can evolve within chronic infected individuals. Here's one study by Choi et al. Published in the New England Journal last year. A 45-year-old with antiphospholipid syndrome had a 154-day infection that ultimately was fatal. He was treated with remdesivir, an antiviral twice, the Regeneron antibody cocktail. They sequenced the genome of the viruses and they found 8 different spike mutations, including one at position of 501 that is defining mutation of several named variants. And here's another case, a 70-year-old with lymphoma with a 102-day infection, treated with all sorts of therapies. That patient had 6 different virus lineages with different spike mutations. So they're all coexisting, including this 2 amino acid deletion at 69 and 70 that I just mentioned. So a way to look at these chronic infections is that they, by following over the time, they basically forecast what could be selected given sufficient mutation supply. It essentially allows us to do some time travel, just like we look back into time. So just as looking into space is like time travel in the past, looking into chronic infections is like peering into the future. So remember, most of these infections are dead ends, but sometimes, very rarely, they do transmit and give rise to a lineage that has many more mutations, producing variants like Alpha and Omicron. And if you're interested in this concept, I really encourage you to listen to this wonderful Radio Lab episode called COVID Crystal Ball. Okay, so it's not just the chronic infections leading to these long branch lengths that allow us to study how the virus is evolving. This expanded genome surveillance and rapid data sharing has made it easier to infer adaptations. So our startup company that grew out of my lab began sequencing the virus very early on in the pandemic, and unfortunately really nobody took us up on it, or very few people took us up on our offer, but a few did. And Jeremy Camille at LSU Health and Lee Harrison's lab here at Pitt UPMC worked with us to sequence the viruses that we could from the resources that we had, and they both deserve a ton of credit for being innovators here. And so we sequenced several thousand samples, and in looking at those samples and looking at their genomes and depositing them quickly into the global database, we noticed something. And so here's just a transcript of a late night email from Jeremy to me. Preliminary analyses suggest there may be a few interesting variants in the latest 367 sequences you just finished and uploaded to the box. I'm seeing ones with spike P681H, Q677H, and Q677P alongside unique constellations of other clade or lineage defining polymorphisms. My suspicion is there could be just as well USA homegrown variants that could be competing fine against the B11 lineage, which then became named Alpha, perhaps explaining why it hasn't begun to take over yet here in the United States. And so I quickly took a look at the data and then estimated the frequency of the genotypes in our data sets. And then I went to the global database and found not only were these viruses increasing in our samples, they were increasing in other people's samples. And just over that weekend, we had an email exchange with Daryl Dahman and Daryl Dinwiddie from New Mexico Health Sciences, who noted a surge in mutations at position 677 in their samples as well. And so it was at this point we thought we were onto something. And so I reached out to Emma Hodcroft, who is one of the key drivers of that wonderful NextStrain site and the Covariant site. And she agreed to join us and help assess the phylogenetic independence of these lineages. And over the next week, we discovered not 2 lineages, but 7 new lineages. We wrote this up and posted it, thinking that it was important. And then shortly thereafter, it became an media storm. And that's sort of what this altmetrics shows. We had a number of attentions. And so the major take homes of this study is that we found 7 new variant lineages arising in the United States carrying mutations at the 677 site. We named these for regional birds instead of the states where they were first detected to sort of avoid stigmatizing, say, Louisiana or the upper Midwest or New Mexico. This 677 site is only 4 residues away from the polybasic furin cleavage site that's critical for cleavage for the spike protein to enter our cells. And now moving on a year later, even though those mutations were part of the old alpha lineage, these mutations continue to rise and spread within new lineage because they continue to evolve within the alpha, delta, and omicron lineages. And these lineages continue to grow in frequency. And so here are 2 global phylogenies that show the actual frequencies of 677, which are relatively rare. But then on the right, when we filter only for lineages that have a mutation, you can see that they recurrently evolve convergently. So this phenomenon of convergent evolution happens at other sites as well. Why we think this is important is because we've since shown in collaboration with Ben-Hur Lee's labs at Mount Sinai that they alter the protease activity acting on the furin cleavage site and improve the essential processing of the spike variant, producing larger syncytia that improve viral titers and spread. So with all of this work, I want to acknowledge a huge collaborative effort of basic researchers, physician scientists, epidemiologists, and virologists to help us tell the story. Jeremy Camille and John Bancheri from LSU, Emma Hodcroft from Bern, Daryl Dahman and Daryl Dinwiddie of New Mexico, Ben-Hur Lee's laboratory, and many others in the open science community. Of course, Dan Snyder, my partner at MIGS. And I also want to thank my lab here at the University of Pittsburgh, and thank you for your attention.